All right, I know folks are still logging in. We do have a packed agenda today, so we will just get started with some introductions. Welcome, everyone, and thanks for coming to session 2 of the Acadia Science Symposium, the first time we've been able to have session 2 or more than one session since we are remote or doing this virtually this year. I'm Nick Fisicelli, President and CEO with Schoodic Institute at Acadia National Park. We are a nonprofit partner of the National Park Service in science and learning. A core role for us, and I think as exemplified here through the Acadia Science Symposium, is to help facilitate science connections and collaborations. Today's topic is monitoring change, a topic near and dear to me, and I think near and dear to everyone that's joining today, whether as a scientist, as a manager, or as an observer of nature. We know change is ongoing and accelerating, and we need people out there, both professional scientists, citizen scientists, helping to document and understand and respond to these changes. So thank you all for joining, and big thanks to the team at Schoodic Institute and at Acadia National Park for all the hard work in creating today's session, and also big thanks to today's speakers as well. And again, we have a wonderful packed agenda, so I'm going to hand it right over to Abe Miller-Rushing, Science Coordinator at Acadia, to fully introduce today and get us kicked off.
Hey, and thank you, Nick. I really appreciate that. And yes, welcome everybody to the second session. This is actually the most requested topic. We've asked our research community what topic folks wanted to hear about, and monitoring change was the one that got far and away the most requests. And so hopefully, well, I think we have a really good program and you'll hear some really good stuff today. Before I hand it over to the speakers, I just want to go through a handful of housekeeping things. So please keep yourself muted, except in the breakout groups, or if you're called on during to ask a question. So I'll try to remind you if I hear anybody unmuted, but please keep yourself muted. It'll also, please update your name in Zoom. So on your little, you can, if you hover over your little picture or whatever, you can click on the ... on the top right, and it should give you ability to rename yourself. Well, that'll help us assign you to breakout groups to correspond with the things that you said you were interested in when you registered. Feel free to also add your pronouns if you'd like there as well. Using video is completely optional and up to you today. And throughout the sessions, please ask questions in the chat. So you can use the chat function, ask questions at any time. We're going to ask each speaker will get to respond to one question after their talk, done their initial talk. They'll each be talking for about 10 minutes. They'll get a chance to answer one question as we transition between speakers. And then we'll hold others till the very end, to the breakout groups and the full group discussion. But we'll use that chat as a catalog of questions as they come up. So feel free to use it as they come. Finally, thank you for your patience with us. This is the biggest change. We've been doing the Science Symposium now for the Acadia National Park Science Symposium for about 9 or 10 years now. This is far and away the biggest change we've had, and we appreciate you being patient with us as we kind of make our change and learn doing these virtual meetings and whatnot. We've been trying to solicit feedback from you and respond to that. And after today's session, let us know what you like, what you don't, and what you think we can change going forward. You'll get an email afterwards with links to some of today's, to the recording of today's talk and some other information. And feel free to respond to that with your comments and suggestions. With that, I'm going to hand it over to our first speaker today, Bick Wheeler, who works for the Park Service as our wildlife biologist here in Acadia National Park. And he's going to open up with a talk about wildlife populations in Acadia and how they're changing.
Sorry, I feel like my monitors are reproducing here, so let's just figure out how to get everything up. There we go. Is that working?
Yes, perfect.
Super. Thanks a lot. Thanks a lot, Abe. So the subject here is wildlife at Acadia, which is extremely broad, and so I ask for your patience to come along with this more aesthetically pleasing, I hope, sort of approach to this presentation. But I personally take an intellectual and intrinsic appeal to nature and wildlife. I'm also eager to inspire others to embrace that love of beauty and wonder. At Acadia, as we're moving into the future, we're trying to endeavor to use science as discovery to build a knowledge base, bridge knowledge gaps. So it's my working hypothesis that this approach will prepare us to better implement science into both foreseen and unforeseen applications. So when I went for a hike with the superintendent, Kevin Schneider, to discuss the future of the wildlife program at Acadia, he asked me, what can I do for you, Vic? What do you need to be successful? And the obvious answer absolutely would have been more staffing, more money, equipment, space, maybe a few political elbows here and there. But instead, I asked for his patience, support, for these instances where we're trying things a little differently and those inevitable failures that come from that. I ask that we embrace those failures as epic failures that lead us to a better next try. Granted, staffing, money, equipment, and space would absolutely be accepted, but it's our ability to fail that allows us to innovate, to question, and to take bold risks. So the direction we plan to take wildlife monitoring at Acadia is with a more focused, in-depth approach to methodologies that have higher sensitivity to change, can drive hypothesis testing studies, and have applications beyond Acadia's borders. Animals move. It's a risk, absolutely, to intentionally reduce breadth and choose depth in our monitoring and studies. We'll be leaving enormous holes in the ideal circumstances of monitoring. But I hope, and this is where we all come in and come together, that through our partnerships, we can make it happen, because the Park Service can't do it alone. So as an example of some basic research with some unforeseen applications is our work with myotic bats. These are through partnerships between the National Park Service, Biodiversity Research Center, Biodiversity Research Institute, Schoodic Institute, and University of Maine at Orono. And then, of course, Acadia's neighbors, because bats fly across political borders, right? So in the past decade, resident bat populations, in particular, 3 species of the genus Myotis, have been plagued by a devastating disease, white nose syndrome. This is a fungal disease that causes mass mortality in hibernation sites. It was first detected in New York State in 2006, and populations in Acadia and range-wide have plummeted significantly. Since 2009, before white nose syndrome arrived, who cared dearly about the bats and Acadia took it upon themselves to further the knowledge base of behavior, species interaction, and natural history of bats in this place. The work looked at 2 critical times in the bat's life cycles: hibernation, which here is a photo of a tricolored bat hibernating, and then also maternity colonies, where females birth young and raise young in social communal settings. Through this work, the literature, though, it indicates that each of these three species of Myotis prefer a different roosting or roost type of structure. One species supposedly roosts in trees, another in buildings, and another in rocks. But in a closer look at Acadia, a closer look at these maternal colonies, the data is mounting that bats are not reading the textbooks. And so during the critical time of the summer, when females raise pups, they're using all types of roosts, including the rock piles and cracks characteristic of the mountains in Acadia, the trees across the landscape, and the buildings both in the park and the surrounding communities. As if breaking the mold wasn't enough, they're using these different strategies in different types of roost structures. So we're beginning to suspect that the eastern small-footed bats use naturally occurring rock roosts, and they use it in a manner where they utilize a network of many different roosts, moving between those roosts, sometimes nightly. Whereas eastern small-footed bat colonies in buildings tend to rely on the same roost throughout rearing pups. It's these fission and fusion behaviors that raise a lot of questions, including transmission rates of pseudogymnastic destructans, which is the causative agent of white nose syndrome, or the long-term viability of the maternity colonies to continue to successfully raise those young. As interesting and fascinating as these findings may be, they do seem a bit intellectual. And how can that be actionable for a little park on the coast? So let's put it into context. As it turns out, Acadia has a series of historic structures, and they all have a long list of overdue restoration projects. The park has fortunately secured funding to make long needed improvements on a few of these structures. And incidentally, these structures are some of the very same that we've been studying the different strategies of roosting behavior in Myotis bats. These basic studies of putting a radio transmitter on a bat and literally not knowing where it would take us, we can now see unforeseen applications. It has now allowed us to directly apply knowledge of these bats and the way they use buildings and show us how extremely important these maternity roosting sites are for three species of threatened and endangered bats. It also shows us the components of these buildings that are highly sensitive and critical to maintain the qualities for these maternity roosts. It's through our partnerships with staff at Acadia and the Historic Preservation Training Center, which is a branch of the Park Service, to conduct this restoration work. We will both preserve the historic structure, which is important as a cultural resource, but also as a structure, it is important to maintain it to be continued resource for bats. So what we intend to do is not just limit the impact of these restoration projects to bats, but also maximize the conservation. So here's another area where we hope to maximize this kind of depth of monitoring and the unforeseen application. Some of the most iconic wildlife at Acadia and some of the best studied are birds. And with that knowledge base, we can go deeper rather than just scratching the surface. A true cross-cutting group of vertebrates that serves as inspiration and source of inquiry. Birds have broad appeal and charisma that captivates the public and the scientific community alike. Another reason birds and monitoring have strong relevance at Acadia is because they're an appropriate model organism for monitoring and studying long-term change. They're long-lived, highly mobile, and at least for now, still abundant. And birds are extremely diverse, at least for vertebrates, and utilize varied habitats. So birds face enormous threats, with many populations at a tipping point. Just in North America since 1970, 30% or 3 billion birds total have been lost. It's staggering. It's the challenges and phenomena concerning this highly diverse group of animals that the application of our potential discovery for monitoring efforts are unknown, at least at this time. As you'll see in the next few presentations, really well-crafted, creative study design, resourcefulness, foresight, and partnerships, investments in the right efforts right now have enormous return later on. Threats to wildlife populations in this era of exceptional change include disease, climate, shifts in species interaction, subtle but intense synergistic effects. Many wildlife in Acadia are highly mobile. We do not need to just keep them alive. It's a much bigger task where we need to actually convince them to stay. These are some of the challenges that bats are facing. And they're highly mobile, but they have high site fidelity. So keeping the resources sufficient and buffering change dramatically builds resiliency in populations. All the wildlife in Acadia have intrinsic value. But in order to have useful discovery in our science, what we need to do is focus on only some groups. Rare animals like bats, common birds, and those with unique stories in Acadia like peregrine falcons or sea-run brook trout are the highest value for the park to focus on right now. Freshwater mussels and butterflies are absolutely important, but we're going to need help. And so we'd like to recognize the amazing creativity and passion within the people of this virtual room, as well as those who we have not yet reached to. It's what I ask is that through our creative partnerships, we take this journey together of failures, successes, to be inspired, explore, and remember endless forms most beautiful and most wonderful have been and are being evolved. Thank you.
That was great. Thanks a bunch, Vic. Feel free to ask any questions in the chat. And as we have time for just one question as we transition to our next speaker. But Camilla, I'll let you load up. You can go ahead and get yours started. If we don't have any questions for Vic, or if you're formulating them now, you can still put them in the chat and we'll get back to them during the breakout groups and in the full group discussion. All right. Well, I think we'll go ahead and move on so we can get through and have more time for discussion. Thank you, Vic. And so our next speaker is going to be Camilla Syrup, who's the plant ecologist for working with our inventory and monitoring network and runs the forest health monitoring throughout a lot of the eastern national parks. And she's going to talk about forest monitoring here in Acadia. And she's also a grad student at the University of Maine. So she's very, very busy. All right, thanks a bunch, Camilla. I'll let you take it from here.
Thanks, Eve. Hi, everyone. Like I said, my name's Camilla Syrup, and I'll be talking about forest monitoring in Acadia and revisiting historic inventory plots. So just a quick outline of the presentation. Briefly going to talk about who I work for, and I'll talk about our forest monitoring in Acadia. But the bulk of the presentation will be about current work resampling historic inventory plots to supplement our monitoring. So I work for the National Park Service's Inventory and Monitoring Division. IMD is made up of 32 networks and provides baseline inventories and long-term monitoring of national resources to support park management. Specifically, I work for the Northeast Temperate Network, which supports parks from Maine to New Jersey. And I am based here in Acadia. So we have been monitoring forests in Acadia since 2006. We have 176 randomly located
I heard you
plots.
crying.
And at this point, they've been sampled at least 4 times. And so I've listed just a few of the ways in which we've used our monitoring data over the years. If you'd like to know more about any of these topics, please speak to me or Kate Miller or visit our website. But I don't have time to get into it today. But we do have the most extensive network of forest monitoring plots in Acadia, and 14 years of monitoring data is a long time, but that's still less than 2 decades of information. So wouldn't it be nice if we could look further back in time? So enter the oldest forest plots on the coast of Maine. They were set up in 1959, over 60 years ago, by Ronald Davis, who is now a professor emeritus at UMaine. But in the 1950s, he was a seasonal park ranger at Acadia and decided to do his doctorate on coastal spruce fir. And he was the first to document the extent of spruce along the Maine coast, which is a local range extension enabled by the maritime climate, meaning the cooler summers, warmer winters, and higher moisture availability. So he identified stands of mature spruce up and down the coast and established forest inventory plots. And 5 of his red spruce sites are in Acadia on MDI. So the sites are located on Pematic Mountain, Otter Point, Blackwoods, Beech Cliff, and Western Mountain. These are just the red spruce sites. And this 60-year time frame could tell us a lot about coastal spruce forest dynamics. Davis's mature spruce sites may be what the rest of Acadia's young spruce forest will look like as they age. So learning as much as we can about how these sites respond to climate and disturbance could help us predict how the rest of the spruce in Acadia will respond. And maybe we should talk about why spruce. Spruce is the most common tree species in Acadia. That's why we're so interested in it. And it's predicted to do very poorly as the climate warms and changes. However, there's evidence that the coast, the Maine coast, acted as a refugium for spruce during the mid-Holocene warming period. So spruce persisted on the coast of Maine when it disappeared from central and northern Maine. So these plots are a great opportunity to deepen our understanding of coastal spruce. Unfortunately, Davis did not permanently mismark his plots, but he did publish site maps. So this is one of his site maps from Beech Cliff in particular. This black rectangle is where he focused his observations, and that's where we wanted to reestablish a monitoring plot. So we geolocated the corners, but these are hand-drawn maps, and they're not completely accurate. But fortunately, Davis provided some other clues, and on this map in particular, he pointed out this boulder. So when we were out there trying to locate the site, we were hoping to find a distinctive boulder that would confirm that we were in the right place. And we found it. This is a real victory and a great looking boulder. So last summer was certainly different, but we did manage to complete some fieldwork. We set up plots at 4 of the sites. We mapped and measured trees and saplings, and we did an inventory of coarse weed debris, and we took increment cores from 30 trees at each site. Next summer, hopefully, we will assess seedlings and ground cover metrics and establish a 5th plot at the Western Mountain site. So this is what the tree maps look per site. So each dot is a tree, and the size of the dot corresponds to the tree's diameter. And you can see there's quite a lot of variation in tree density and size across the 4 sites. They look quite different. In a moment, I'm going to show you some preliminary results of the tree distributions for each site, but I just have to go over a few things before we get there. First, the age distributions I'm going to show you are from the 30 tree cores we took from each site. The trees were randomly chosen and stratified by diameter, so they're representative of the site as a whole. They're not just, the tallest or biggest trees. And second, I'm going to be talking about recruitment age, which is not the same as the total age of the tree. Recruitment age is the years since the tree reached coring height or sapling size, and it doesn't include the years it's spent as a seedling. And finally, I just want to reemphasize these are preliminary, and we won't have a complete picture of each of the sites until we complete all of our analyses. So without further ado, these are the age distributions for red spruce at each of the sites. On the x-axis, you have the decade each tree was recruited. On the y-axis is the number of trees recruited per decade. So we'll start with Pematic Mountain in the upper left. So for this site, all the trees recruited in one pulse. They're all kind of clustered around the 1880s, 1890s. All the trees we record are about the same age. And these trees likely grew in after a harvest. And it's sort of a one cohort stand. If we go to Beech Mountain to the right, the trees are sort of clustered around 1880 and 1890, but it's a much messier picture. There's some trees that grew to sapling height in the decades previous. There's some trees growing in the decades after, and we don't really know what's going on here yet. And looking more closely at the tree cores will give us a better idea of the history of that particular stand. If we go to the bottom left to Blackwoods, this site's really interesting. There's 3 distinct cohorts at this site. The main pulse of recruitment and most of the trees that we record are from the 1880s and 1890s. Again, this does look like a response after a harvest, but in this case, there are some trees that are older from 1810s to 1830s. And this could have been that they were too small during the harvest and they were not cut down, or perhaps the site was used as rangeland and these were, solitary trees in a field. We can actually reconstruct the diameter of these trees back in 1920 and 1930 and see if we can get any more hints to what was going on at that site. And the last cohort here in the 1920s and 1930s, this is likely a response after a spruce budworm outbreak. There's a documented spruce budworm outbreak in the early 1920s, and likely that opened up the canopy and allowed a few more trees to make it to sapling height. Finally, Otter Point site, it also appears to have 3 cohorts of trees. This is a different situation. The main pulse of sapling recruits occurred in 1960s and 70s, which is much later. And we know that there was a really severe windstorm in 1961 or 62, just a few years after Davis sampled this site, and it blew down most of the trees. And so this is a response to a fairly recent disturbance, and it's going to be really interesting to look closer at that site in particular, since we have data immediately prior to a major disturbance. So just to wrap up the primary objectives and next steps for this project, number 1 is to quantify the 60 years of change in forest structure and composition at the Davis sites. When we'll extend these insights from just the Davis sites to that rest of Acadia National Park through comparisons with the randomly located NETM plots. And we hope to generate management recommendations for coastal spruce as part of this effort. The second big piece of the project is to investigate the climate sensitivity of spruce. And what I mean by climate sensitivity is how does spruce growth respond to climate variables like temperature or precipitation? And we want to look at this at different scales. So at the local level, do spruce respond differently to climate based on site conditions like aspect or soil depth? And what about at the regional level? Does coastal spruce respond to climate differently than high elevation or inland spruce in other parts of New England? So answering these questions will help us predict how coastal spruce will respond to climate change. I think that's all my time. I just want to thank the Northeast Temperate Network and Scudic Institute for funding this research. Sean Fravor and Jay Wason are the primary investigators on this project, and Kate Miller is the lead for the Northeast Temperate Network Forest Monitoring Program. Thanks. I think maybe we can have one question.
Yeah. So one of the questions is kind of a basic one here for you. How do you determine recruitment age?
Yes. So.
And then Jasmine, you can go ahead and load your slide.
When you take a tree core, you measure, you count every tree ring and measure the ring widths, and then you you date each ring based on, this is a little bit more than I can get into, but you can assign dates to each ring. And so the recruitment age is actually when you hit the pith. So it's the earliest ring on your tree core if you took it at sapling height, which in this case was 1.37 meters. I can probably explain that better in the breakout group. Yes, sorry, hope that made sense.
No, I think that was good. Yeah, and I'm guessing that there will be more questions about aging the trees and whatnot. But yeah, that was great. Thank you. So our next speaker will be Jasmine Saros, who's going to be talking about changing lake ecology in Maine. And Jasmine is the Associate Director for the Climate Change Institute at the University of Maine. Take it away,
Thanks,
Jasmine.
Abe. Thank you. So as Abe said, I'll be talking about changing lake ecology in Acadia today. And so just to start off with the usual question of why do we study lakes? And really one of the key reasons that we study lakes is because they're of great value to us in a lot of different ways. And so we can think about some of the value in particular of Acadia's lakes and thinking about their cultural value with recreation and aesthetic features. We'll also see today some of the scientific and ecological value of these systems. We also know, of course, there's an economic value in the realm of tourism. And in Acadia, some of the lakes serve as drinking water sources as well. And so these are drinking water sources to the towns on MDI and also, of course, to the visitors to the park. And so similar to what we see in a lot of Maine lakes here are serving as drinking water sources. But one of the key reasons as well that we study lake ecosystems is because they're sentinels of environmental change. And what we mean by that is that they're often the first system on a landscape to exhibit measurable changes in response to some sort of driver or disturbance. And so when we think about drivers, this could be larger scale drivers such as climate change, could also be changes in land use as well. And so compared to what we see, say, on the landscape itself, we might actually be able to detect changes in a lake first. And that's largely because lakes serve as integrators of information of what's happening on their watersheds, or in other words, on the landscape around lakes, and also in the airshed or the air above lakes as well. So they're materials that can flush in from the landscape and from the atmosphere that basically all kind of get integrated into lakes. And that's one of the key reasons why we tend to see change here first. So along those lines then, I want to talk today about a couple different lake responses in particular. I'm going to focus quite a bit on water clarity, largely because this is not only an important feature aesthetically, but also ecologically. Water clarity has a big influence on habitat quality in lake ecosystems. And I'll show a little bit as well about some of the drivers of changes in water clarity. The water clarity data and some of the associated chemical data that go with that come from the long-term monitoring program by the National Park Service. So it's a really great data set that has enabled us to identify some changes that are going on in lakes in the park. I'm also going to talk just a little bit about water temperatures and ice cover. This is work that really we've only been focused on for about a decade now. And so I can't really tell you too much about trends, but I'll just show you some of the approaches that we're using and some thoughts as far as like where we're going with that type of research. So thinking about water clarity, and so how clear a lake system is, one of the key ways that we measure this is using a Secchi disk. And so a Secchi disk is basically, if you're not familiar with it, it's a disk that we lower down through the water column and we look to see where does it disappear. And so it's a metric that we use basically to compare across systems and also within the same system over time. And so these plots are showing you different lakes in the park, and we're looking at the Secchi depth. And limnologists, lake ecologists, we like to plot things sometimes so that The surface of the lake is at the top of the plot here. So we're at 0 depth, and then we're moving down into the lake on this diagram. And what you're seeing here is that basically as you're up in the shallower part of the lake, if a Secchi reading is up in the shallower part here, it's a less transparent reading. And then as you go down deeper, if you can see your Secchi disk deeper, of course, it means the lake is more transparent. So this is showing you what's been happening across several of these lakes since about 1980 to 2008. And we're looking at Jordan. It shows the most striking example, I'll say. And what we see is that from about 1980 to 1995, water clarity was increasing in Jordan Pond. And then from about 1995 to 2008, water clarity was decreasing. And you can see a similar pattern as you look across these different lakes. And so a coherent change where in more recent times, we've been seeing a decrease in water clarity. So to think about why that has been happening, you want to think about what affects water clarity. And there's really two major materials that I'll talk about today, and that is one being chlorophyll, which is present in algae, and that imparts this sort of greenish color, of course. So as we see blooms of algae, you can see a decline in water clarity. But another material that contributes to this is dissolved organic carbon. And so you can see we're looking at a series here of increasing dissolved organic carbon, so the water becoming browner as you increase this concentration. And this is what a Secchi would look like in this browner, higher DOC water. Dissolved organic carbon comes from the watershed. There is some produced in lakes too, but in main lakes and in Acadia lakes, most of it is coming from outside of the lake. And it's a natural material. It comes from the decomposition of plant or animal material. And as it's produced on the watershed, then it can undergo a lot of change and transformation, but eventually it's going to find its way into surface waters in the area. The amount of precipitation, the timing of precipitation, the chemistry of precipitation, these are all things that can influence how much of this organic material is going to make it into surface waters. And so if we look at what's been happening in terms of dissolved organic carbon and also chlorophyll in these same lakes in Acadia that I was showing you the water clarity trends in, now we're just looking from 1995 to 2008, so the period of time where we were seeing decreasing water clarity. And DOC is shown in the dark circles and the black lines here. You can see that that material has been increasing over the same time frame where we saw the decrease in water clarity. And you see a similar trend as you look across the different lakes here. Whereas if you think about chlorophyll, this is the open circles, we're not seeing a trend in terms of what's been happening with chlorophyll. And so we think the changes in water clarity are being driven by an increase in dissolved organic carbon across these lakes. This is important not only for water clarity, but DOC is actually considered one of the master regulators of aquatic ecosystem structure and function. So it's a very important material when we think about the ecology of lake systems. I talked about water clarity. So that also is another way of thinking about it is thinking about the amount of light available in the system, which is important for photosynthesis and production. But we also know that DOC affects the temperature structure of lakes because it affects heat retention in the systems. DOC also plays an important role in affecting nutrient availability, both directly and also indirectly. It can affect pH, and it can also affect bacterial communities. And that's a big concern when we're thinking about drinking water lakes. If we have more DOC, it can facilitate the growth of bacteria, but not always, but in some cases it does. So why do we think DOC has been changing across these lake systems from about 1995 to 2008? Well, it turns out we actually think it's a positive sign. We think it's a sign of recovery from sulfur deposition. And so if you look at what's been happening with sulfate deposition broadly across the country, but we can kind of think, focus in on the Northeast. Here's a snapshot from 1994 from the National Atmospheric Deposition Program. And then we can just sort of move through time here, and you can see how it's been declining. And we know that sulfur deposition affects the pH of soils, and that can affect how well soils either retain dissolved organic carbon or release it into surface waters. And this recovery from acid deposition, we think is essentially leading to a release of some of that organic material into lake systems. And I can talk about this more during the breakout session if people are interested in that. But we do think this is actually a good sign and a sign of some recovery from acid deposition. I showed you the trends in water clarity up to 2008. So I wanted to show you an update of what's been happening since then. So this line here is about 2008. And what you can see is that those changes in water clarity have actually stabilized. So we're seeing still quite a bit of variability, as you always would in water clarity, but no particular trend since 2008. Okay, as I said, I want to just talk a little bit about what's been happening with temperature and ice cover. And this is something we've really only been looking at just recently over this last decade, at least from the data that I've seen. But I can say more broadly, thinking about the timing of spring ice out on lakes in New England, there's some nice work from Hodgkins et al. Showing that since about the mid 1800s to 2000, we've been seeing that the timing of ice out has been becoming earlier. And if we look in Acadia, I can just show you a little bit of data from Jordan Pond, just to show you what the ice cover looks like and just how variable it is, as you might expect in lakes along the coast. You know, some years where Jordan Pond, for example, has it was never completely frozen over, and other times where we had a longer period of ice cover. And interestingly, if you just look at the timing of ice out, which would be the end of the bar here, it's quite variable on Jordan Pond. And so we're trying to take advantage of this to sort of look at what happens in years with early ice out versus late ice out. And ecologically then, what do we see shaping up in lakes as a consequence of these changing ice out dates, or the variation in ice out dates? We're also looking quite a bit more at temperature in these lake systems. And this just shows you a few different plots of over the course of the open water season, what the temperature profiles look like, in this case, again, in Jordan Pond. And just to say that we're using some high frequency sensors now to really track these temperature changes and compare them across years and think about what shapes the temperature structure of these lakes over time. And then finally, some research from my master's student, Matt Farragher. He's tracking additional changes with high resolution sensors. So he goes out on the ice in the wintertime and deploys these sensor strings where he's measuring temperature, dissolved oxygen, and light under the ice, and then allowing those sensors to stay in over the open water season as well. So he can see what kind of a role that under ice period plays in shaping up what we see during the open water season. And that's just some of the data here, looking at dissolved oxygen in surface waters and deeper waters in a couple different lakes around the park. So hopefully we'll be able to talk more about that in the not too distant future. So with that, I'll just say I want to thank Bill Gawley for the long-term data and also for continued collaboration, and also say a thank you to Friends of Acadia for their continued support of this work.
Great, thank you, Jasmine. That was terrific. And I'll just point people that at the beginning of the chat, I put a link into the to where you can find the posters and infographics and a video that'll be that are a part of the symposium. And actually, Matt has a poster there, Matt Farragher, who Jasmine mentioned. One quick question for you, Jasmine, that came up is: are you working with the volunteer lake, or the Lake Stewards of Maine, or volunteer lake monitoring program?
Not specifically for this work, because the park has such a great database at this point. But yeah, for other lakes in Maine, that's a great resource.
Yeah, for sure.
Yeah.
We're really lucky to have a very strong culture statewide of volunteerism in science.
Yeah, absolutely.
Well, thank you, Jasmine. I think we're gonna move on to the last of our speakers. And we did know that we would likely run into the breakout groups a little bit. So this will be our last speaker, and then we'll move into the breakout groups after this. So the last talk is Hannah Weber, who's the Director of Marine Ecology at the Scudic Institute, and she's going to be talking about intertidal monitoring in Acadia. All yours, Hannah.
Cool. Thanks, Abe. Thanks to the symposium planners, by the way, for putting this on. I'm happy to be here today, and thanks to my other speakers. You guys are awesome. Anyway, I'll be talking about monitoring in the intertidal zone. Here, of course, is the quick talk outline. We'll do a quick orientation into the intertidal, and then the 3 Ys of intertidal monitoring, the 4 S's of intertidal monitoring. I'll be quizzing you later on all this, and then monitoring gaps in future directions. First, of course, the quick orientation. And let's see, great, got that to work. We, of course, at Acadia sit at the edge of the ocean, and the ocean sloshes up and sloshes back down the shore twice a day. And there's about a 12-foot vertical difference between that high tide, that slosh up, and the low tide, that slosh down in Frenchman Bay. And in that 12-ish feet exists the intertidal, that world of organisms that survive being in the ocean for half of the time and out of the ocean half of the time. The intertidal here at Acadia, of course, is comprised of beach, salt marsh, mudflat, and rocky intertidal. And the rest of this talk is largely going to be on the rocky intertidal, and I'll sort of swing back around to mudflat at the end. There's our orientation, and here's our 3 Ys. Why monitor? So 1, bad things happen to good ecosystems, or monitoring provides a baseline against which we can compare the effects of the bad things. 2, is this normal? Or monitoring helps us set the parameters of the realm of normal. And 3, the lament of all things, why this? Why now? Or monitoring is attending to pattern and therefore acts as a launch pad to exploring process and mechanism, or as Vic said earlier, drives hypothesis testing. So first, bad things happen to good ecosystems. This is historical. In 1989, the Exxon Valdez hit a reef and spilled 11 million gallons of North Slope crude into Prince William Sound. There were no pre-spill data to look at post-spill comparisons. So, it was very difficult to understand when ecological integrity had been regained after the spill. That was a bad thing. Out of the Exxon Valdez spill came networked intertidal monitoring. So setting up indicators that could be measured periodically and systematically to assess change. And that networked intertidal monitoring approach included, and still includes, the Northeast Temperate Network monitoring of the intertidal. This brings me to my second point. Is this normal? Is this normal? So starting in 2007, National Park Service personnel and trained volunteers have collected data from 9 sites at Acadia and Boston Harbor Island, studying 6 community types within the rocky intertidal. The data are collected once a year in the late spring, early summer. The monitoring effort collects data for key metrics representing the ecological health of the intertidal zone. And over time, it does provide that indication of trend in these metrics. Monitoring is not intended to be comprehensive assessment of the whole ecosystem or the factors contributing to change in condition. So again, are we ever going to have like a Valdez in Bar Harbor? I don't think so, I hope not, but there are other pressures, and we can use these data to think about other drivers of change. Anyway, again, with these data, we can evaluate trends through time. Again, is this normal? And normal is often change. So is this rate and pattern of change normal? And here's an example of that. Here we have on the top right, barnacles. Semibalanus balanoides, an animal, and on the lower right, we have Fucus vesiculosus, a macroalgae. So both of these can be dominant in the intertidal zone in particular areas. And what we see Ed Sharon here over on the left doing is taking a picture of a barnacle plot. Barnacle plot is one of the net and monitoring tools. The plots are photographed every year, and then a determination is made about how much space is taken up by barnacles and how much space is taken up with other things. So here's some preliminary data analysis of those barnacle plots from 2007 onward. These are data given to me by Hannah Middlestat at UMaine, and these are draft data I want to mention. So we have barnacles, the gray line, the sort of tan line is barnacles, the gray line is sort of bare rock, and then the green line is fucus. For those of you who might be colorblind and these might not be good colors for you, the upper line is barnacles and the two lower lines are fucus and bare rock. These are from Otter Point and Schoodic Point. We can see barnacles basically dominate the cover, but here's what I want you to look at. At Ship Harbor, fucus, again, this macroalgae is taking over the barnacle plots. And again, is this normal? And it actually could be normal. In intertidal succession, it's not uncommon for barnacles to sort of move in initially to a disturbed area and then for fucus to take over the barnacles later on in succession. However, it could also be indicating something else, which sort of brings me to my third point of why monitor. Again, why this? Why now? Monitoring can be this launch pad for digging into process and mechanism, or for generating hypothesis-driven work. Monitoring is not necessarily, again, about process and mechanism. We don't know why the barnacles are declining or why fucus is increasing. So this bumps the monitoring outcome into sort of a launch point for research. We can design studies, add additional data to help unpack process and mechanism. We can unpack that why this, why now. So that's the three whys and the four S's now: scope, scale, sensitivity, and site of intertidal monitoring. So net-in monitoring is focused on the whole intertidal. Similar in scope, but a little different in scale, is the Northeastern Coastal Stations Alliance intertidal monitoring with a site on the Schoodic Peninsula. I've shown the net-in sites here sort of for reference. We have the net-in sites, of course, in Acadia and Boston Harbor Islands, and the Nexa sites sort of nest within and complement that array, with sites throughout the Gulf of Maine at coastal Maine field stations. So similar in scope, different in scale, Nexa allows us to sort of capture things like the different temperature signatures across the Gulf of Maine, something that Nexa just, net-in doesn't really allow us to do. So we have sort of the cooler eastern Gulf sites and the warmer western Gulf sites, which is what's shown here on the right. However, Nexa doesn't allow us to capture that intra-Acadia differences like the barnacle fucus story. I should mention that sort of net and Nexa data are collected once yearly, and that kind of precludes understanding within year changes or phenology changes. So a third monitoring effort going on in the park, in addition to net and Nexa, is the Signs of the Seasons Rockweed Phenology monitoring. This monitoring is conducted by trained volunteers. Currently, there's one location in the park, but there are also multiple locations around Maine and New Hampshire. Signs of the Seasons is a citizen science phenology program spearheaded by the UMaine Cooperative Extension and is largely coincident with the National Phenology Network efforts. Rockweed is a foundation species in the intertidal and releases gametes when the sea surface temperature hits 6 degrees C and sort of maxes out when the sea surface temperature hits 10 degrees C. So the release of gametes is this springtime pulse of high protein food into the intertidal and nearshore system. And we're monitoring sort of the gamete release and sea surface temperature. So we know sea surface temperatures is changing in the Gulf of Maine as well as globally. And so how is that one specific driver affecting the intertidal, specifically the phenology of a foundation species? And then are there trophic implications for any shift in the pulse of reproductive materials? So the scope of this monitoring is very specific. It's observation of the reproductive parts of a specific macroalgae as they may be sensitive to a specific pressure, changes in sea surface temperature. So, Netten and Nexa and Signs of the Seasons each have this specificity and scope and scale and sensitivity and sight that are different from each other. But having said that and scaling up, any of these efforts could work at a larger scale. Signs of the Seasons protocol is universal and can be applied anywhere in the global rockweed range, and the data can be comparable. And the data from Netten and Nexa can certainly fit easily into like the MBON pole-to-pole biodiversity effort, or be used for an East Coast, West Coast comparison. So, the 3 Ys, the 4 S's, and now the gaps. So this whole talk was about the rocky intertidal without much reference to the other 3 types of intertidal systems that we have here. So for that, we could either think about what monitoring might look like in those, or invest in monitoring, or to go back to Vic's comment about sort of centering in. We should probably be thinking about whether the rocky intertidal change can be sentinel for these other habitats. Wrapping up, this summer, sort of jumping over to the soft sediment really quickly, or the mudflats, we will be doing an inventory survey. So not monitoring, and that, an inventory is that sort of one-time enumeration. But this may provide us that groundwork for either thinking about sentinel or for thinking about monitoring in the future. So again, for myself and all the speakers, I want to thank the organizers, and I want to thank my fellow speakers. I have learned a lot, and I'm looking forward to the breakouts and the group discussion. So thank you.
Thanks a bunch, Hannah. That was great. That was awesome. It doesn't look like we have any questions right away, but feel free to keep populating the questions. And Catherine, do you have the slides for the breakout, or I can put it up, I think. Anyhow, we're gonna move now into the breakout sessions. The goal of the breakout sessions is for you to talk more with some of the speakers and some of the people who prepared posters or videos for the symposium. And the goal is really to ask those folks questions, but also to network. And so this is our first kind of our next best effort at promoting networking virtually to kind of in these small breakout groups. So do take the conversations where you want to go. Please start with introductions. And we, after you do the introductions, we posed like 3 questions that you could use to help format or kind of as prompts during your discussions. What questions do you have about the presentations, or what surprised you, or what stories do you think are important to share? And after you come back, no need to take copious notes or anything, but when you come back, we'll have a full group discussion, and just be ready and post any comments or questions that you want to pose to the full group. And we'll end with that kind of full group discussion, and particularly with the panel answering questions. All right. So with that, I think we will come back as a group at 1:20, I think. We'll come back as a group for the last 40 minutes of the session. Welcome back, everybody. Well, I think people are still filtering in, so we'll let that happen over the next minute or so as people come back. Feel free to start adding your questions or comments or things that came out of your breakout groups as you bot back. Just throw them into the chat.
Abe, do you want me to share this or let it so we can all see each other?
Yeah, why don't you just let everybody see each other, I think.
Okay.
Be fine. As I think everybody just about is back now. Feel free to please put your questions, any questions that came out of your group or any comments that you want to share with the full group, put them into the chat. And as people are doing that, we can start with some of the questions start this discussion with some of the questions that came up during the presentations before the breakout groups. And to start with, I'm going to go back to Bick, our first speaker. Michael Good mentioned eBird, had a comment about eBird data, and I was just to turn that into a question. How much do you actually work with citizen science data or eBird specifically in the wildlife monitoring, Bick?
Yeah, so I guess there's a lot of levels. Certainly eBird is sort of one of those shining stars for how to implement this type of data gathering and really make it into some incredible, robust findings and apply it across so many scales, which I think is really great. It's not something that I've worked with myself. It's I'm mostly a small data kind of person. But it is really exciting, and as we are in our breakout session, we were talking a lot about technology, and that is one of those pieces of technology. Although my phone is old and broken, you know, it's an amazing piece of technology to put in so many people's pockets. Just, you know, it's amazing. A lot of the work we do, just having a time and place attached to an observation is incredible, and it's incredible gold. And so there's some really great work to be done. I think I have not personally done a lot myself, but I'm excited to jump into it.
No, that's great. And do you mind just explaining just real briefly what eBird is?
Sorry about that. Sure. So eBird is it was developed to look at or to take observations by citizen scientists. And so to take observations by somebody going out birding and just finding, taking those species lists that they take from each day or each site, and then with those, each of those observations into a massive database run by Cornell Lab of Ornithology, those data sets can really be pulled into tracking everything from phenology of when birds are where, migration, and some really, really neat stuff. So a terrible explanation of what it is, but it is very, very cool.
Yeah, I just encourage anybody to go poke around that, but that was great, Bick. You can find it at ebird.org or in the App Store, Google Play Store, whatnot. The next question I'll pose came early on for Camilla. I think it was, well, anyhow, I've lost track of exactly who asked it. But is there a difference between the interior spruce and the coastal trees at the edge of the water? I think that was kind of striking from your presentation, that how localized some of the disturbances were that drove a lot of the recruitment differences across the places.
Yeah, looking at that fine a scale, like on MDI, how, distance from the ocean, could be very challenging with the low sample size. We only have 5 plots. When I was talking about inland spruce, I meant like Bangor area, outside of sort of the maritime range. That's not to say there isn't differences. There's certainly that coastal fog. There's a lot more coastal fog right on the coast. It does cover the entire island, but it's in Southwest Harbor a lot more often than it is up on top of Western Mountain. We'll just need, I think, the NETN plots, coring outside the NETN plots, that we have so many more of them, might be able to answer that question better than the low sample size of Davis plots that we have there. So it's a really interesting question. I think we just would need more data to get at it.
Great. Yeah. Did that answer? I figured it out. So that was Catherine, I think, that asked that question. Did that answer your question, Catherine?
It does. And I guess if there's anyone else in this audience who has even sort of anecdotal or observational things that you've noticed that are different between the forest that's right on the edge of the water, right, versus the interior Mount Desert Island forests. I'm working on an article about island forests, and we at Schoodic have revisited some forest plots on islands around Acadia. And so I've just been thinking about this question a lot, and I'm curious.
So I can jump in just a little bit in case it helps. Generally, like right along the coast, it's white spruce instead of red. So that's kind of the big difference, just like a fringe right along, like where it's rocky going right close to the water. There does tend to be more wind disturbance. If you look at like annual kind of disturbance rate, it tends to be slightly higher right along the coast versus, even just like 100 meters inland or something like that. But beyond that, I don't really know if we have a good answer to that.
Anybody else want to take a stab at that? It's hard to follow Kate and Camilla answering that question, though.
Thank you both.
All right. So the next question, Aaron, you had a question about DOC and chlorophyll. Do you want to ask that? Do you want to unmute yourself and ask that for Jasmine?
I can do that. I can even zoom in here. Hi, Jasmine. Yeah, so I was just curious if there was a strong sort of correlation between DOC and chlorophyll A, particularly in sort of any of the given water bodies. Is that a consistent relationship?
Hey, Aaron. Not really. I would say usually we don't see a relationship between them. And I would say because DOC is, most of it is coming in from the watershed. And then with chlorophyll, we're measuring something that's being generated within the lake. And that chlorophyll can be influenced by DOC in terms of the water clarity. So in other words, light coming to algae. And DOC can also carry some nutrients with it. But there are a lot of other things also that influence chlorophyll. So usually we don't see them tightly coupled. Okay. Sometimes you will, but for the most part, usually not.
Okay, great, thanks. No, that's super helpful.
Okay.
And Bill, you had a question, another question for Camilla. Do you want to unmute yourself and ask that?
Sure, thanks. Yeah, I was just curious because you were talking about the Davis plots, Camilla, and GPSing those sites. Now that they're GPSed, are you looking at satellite imagery or any remote sense data related to those? I know it's a different spatial scale than the plot level, but looking at stand level or larger changes, perhaps depending on what data are available going back in time.
That's a great idea. We are not currently doing that, but that would be a great way to supplement, once we synthesize all of the actual plot changes, to sort of expand that out, or to confirm, if we can, if there is imagery from the time period that we're looking at, the dendrochronology information. It would be a good way to sort of get a second line of confirmation. But that's a great idea. That would be another line to look into.
Just a quick follow-up. It also, I mean, given that you're doing plot level, fine scale measurements, if you have satellite imagery or any other remote sense data, trying to tie in changes you see at the larger remote sense data into the specific areas where you have the plot level, and seeing if there's any type of trends or something like that, maybe potentially being able to extrapolate to other areas. But anyway, it's just, I think there's a bunch of different questions that perhaps you could look into by looking at those data.
Yep, that's a great idea.
Thanks, Bill. And Camilla. I think I'm going to ask one for Hannah. So Hannah, you talked about the fact that things are changing at one of your sites. I've forgotten exactly. I think it was Ship Harbor, maybe, much differently than the other two sites in the intertidal that are being monitored. I think maybe those were the NETN data. And is it strange to see that kind of localized dramatic change in the intertidal community? And also would the monitoring across like different scales that you had help answer what might be going on there?
Thanks for asking. So it was at Ship Harbor, and so we're seeing fucus overtaking barnacles in the barnacle plots, which is not unusual. And so the next step is to look at locations that have the same slope, the same aspect, the same, just sort of like soil conditions, only the same rock, sort of like the same roughness of the rock, et cetera, to see if there are other sites where we are seeing something similar. So to go to the Nexa sites, but also to go back to the Netten sites to see if there are some things that are in common between different sites, showing the same pattern, or if it is just, in Ship Harbor. And again, in Ship Harbor, because, we don't have a history before 2007, it's possible, it's not impossible that there could have been a disturbance, like a large ice scouring event, and that all of the barnacles everything got scraped off. And so we're just seeing a different timeline in terms of succession there than we're seeing elsewhere. And that, where we see barnacles in the other places could be just the end of the line. They're going to stay barnacles, but at Ship Harbor, it would be more natural for that to revert to fucus. One, it could also be that at Ship Harbor, we're seeing some different pressures. There could be a different predator suite coming in, eating barnacles, or a different herbivore suite, leaving that's not eating the fucus. So there's a whole bunch of different reasons. It's not unsurprising to see this at one location, but it does then start begging a bunch of questions that we can start digging into.
Oh, that's awesome. Thank you, Hannah. And Mark, I think you had a question for Jasmine about pH.
Yes, I wanted to know where the data would be available, either in published form or so I'm concerned about two things. I'm concerned about pH variation. Not too long ago, Lakes in Acadia National Park showed episodic events. I'm wondering if that's still going on. And I'm wondering what the current sulfur trends are in Acadia Lakes.
So I always ask Bill Golly for the data. So that's, I think Bill is the best resource. He has the most updated, there he is. So I don't know if Bill wants to address some of that.
I just messaged Mark privately to give him that same advice, but that we do have a few different there are some ways to see some of the data publicly. jpbuoy.com in the summer when the buoy is deployed, that has basically quasi live readings. It's updated twice a day. And I'm not sure if you can actually go in and get, the general public can get cumulative, historic readings, but that's another thing that you could definitely check with me on. As far as our historical monitoring data is concerned, we don't have the database is on IRMA, but it's only downloadable to NPS employees, unfortunately. So we have a variety of reports that are on IRMA, the data store that kind of documents some of those things. And I think Jasmine's advice is best at this point. Contact me directly. In response to your specific question about the general trends, we have noticed corresponding with the decrease in sulfate and nitrate in the atmospheric deposition, precipitation pH has increased markedly since, the 1980s, early 1980s, when we've began monitoring that, which we do on a weekly basis. And it seems to be tying over in the same trends in the lake water chemistry as well, too, that both there seem to be, I'm not sure exactly how significant they are. It kind of varies from, water body to water body, but there's definitely increases in pH and ANC in most of the lakes. We don't have quite as long a record of the streams, but, I would expect to see similar trends, but, we're still not out of the woods as far as threats from episodic acidification anyway.
Did that answer your whole question, or Jasmine, did you want to add anything?
No, I think that was great. I mean, we have a couple simple plots of what's been happening with lake water sulfate that Matt Farragher has done, and basically showing a decline in sulfate concentrations in the lakes over the last, since 1995. But yeah.
And Jill, you had not exactly a question, but a point about comparing Acadia with Canaan Valley. Do you mind explaining what Canaan Valley is and kind of what might be about what you were thinking about that comparison? If you're still around. We might have lost Jill. Okay, we will come back. We'll skip over that one, but I think that's a forest that's actually a wildlife refuge, I believe, in West Virginia. I think it's managed by the Fish and Wildlife Service. That's populated primarily by red spruce communities that are typical of up here in Canada.
Yeah, Abe, I'll just hop in on that one. Yeah, I think, right, Jill was thinking about red spruce sort of at the down towards the southern end of its range, and there are the spruce forests there that are in many ways similar to here, but also, also may have some important differences. So thinking about, are we seeing changes here similar to there or not? And I think that kind of gets at a discussion we were having in the forest group as well about monitoring, certainly here in Acadia, but across the state of Maine and elsewhere, to see, are trends here the local trends, are they similar to what's happening elsewhere or not? What are the factors behind that?
Yeah, well, on that site, That's like would have interesting implications for protecting refugia going forward too, which is something we're thinking about also. How long they last and how they work. Erica, you had a question for Jasmine or Matt, I think.
Yeah, I just wanted to know what kinds of lakes would make the best sentinels for climate change, especially like here in Acadia. Do they have to meet any certain like physical characteristics, like the area, depth, bathymetry? And maybe if you could choose a lake in Acadia that would best like capture that, what would it be, if possible?
I could try to take
Yeah.
a shot at. Okay. Yeah, I think that's a great question. I think I chose lakes in Acadia National Park because they're sort of protected from land use, or I think lakes in national parks in general might have a bit less influence from land use or other contributing factors. So it sort of makes it easier just to study the long-term effects of changing temperature or DOC or sulfate directly without other confounding variables. And I think to address your other point on lake bathymetry or the other lake characteristics, I think if it's a lake that's sort of representative of lakes in that area, if it's an average depth or pretty similar to the other lakes in terms of size or other characteristics, then it seems like it would be a good lake to choose for studying or for extrapolating those changes to other lakes. So I think in Acadia in particular, I think Jordan is a great example of a lake like that, which is perhaps why the buoy is in Jordan Pond, because it's sort of protected from other land use and other anthropogenic influences. So I think that makes it a good lake to study. Jasmine or anybody have any other input?
I think that's great, Matt. I think your point, especially about reducing the effect of other drivers as well. I mean, we know there's always multiple drivers at work, but trying to reduce that as much as you can. And I like your response too about something representative. So if you're thinking about broadly, what might you see in a particular area? And what I could just add to that is part of it, I think, depends on what aspect of climate change you might particularly be interested in. And so when we're thinking about changes being driven by temperature or wind, so basically things that relate to energy influx, these deeper, clearer lakes tend to be more responsive to those types of drivers. And so that's, I think, one of the key reasons why Jordan tends to be so responsive. If you're thinking more about inputs of materials from the watershed, so precipitation driven, I think a lot of lakes are going to be responsive to that. But in terms of the really trying to capture some of the energy driven differences, sometimes the deeper, clearer lakes can be more responsive to that.
That's great.
I think
Bill was going to add some more too.
Go for it.
one more kind of 2 cents to put in. I think we're fortunate in Acadia that we have so many lakes potentially that we are able to study, because it allows us to kind of look at some of those different characteristics. We have sometimes 2 lakes of similar trophic types, but some are totally protected. Others are, adjacent to more anthropogenic threats and things like that. So at times, we're fortunate when we select our study sites that we can kind of pair things off and make sure that we can get all those potential threats and variables into, and collect data simultaneously for all of them. So we're lucky.
Yeah, yeah. I mean, there are a lot of good things that make Acadia. We're spoiled in terms of places to do good research. Yes, and good sentinels for different things. That's great. So, Chris, you had a question about microclimates that looks like it got an upvote here. Do you wanna put a voice to that question?
Sure, and thanks for the upvote. I like that. Yeah, so I had a question about microclimates, and so just to be clear, in case people aren't familiar, microclimates are just these places on the landscape that have small places on the landscape that just have a very different climate than the surrounding region. And so we're all familiar with these, like on a hot day, if you walk under a tree to get shade, that's a microclimate. And so I'm curious, what do we know about microclimates in the various ecosystems in the park? And given all the monitoring and all the people that are out there seeing how things are changing, do we think things are changing differently in these microclimates under climate change? What do we know about that? And that's a question for intertidal, wildlife, forests. I think it applies everywhere.
Yeah, I'll actually open that up. Does anybody want to tackle that one? Speaker or not speaker? No, nobody wants to tackle that one.
Wait, I can sort of maybe add to that, or another way of asking, because I think people know they are, right? They exist. And I think we all know how much weather and climate varies across the island. Certainly, while Nick Fisichella the tree test beds that are located at different locations. So sun exposure and elevation makes a big difference. But I also think, I wonder, and maybe this is a question for Bill, we have the monitoring at McFarland Hill, but I wonder, are there other temperature monitor, just thinking of like temperature and precip, are there other monitoring stations around Mount Desert Island besides the main station at McFarland Hill?
Yeah, currently we've got at our air quality monitoring site at McFarland Hill, we have a pretty full range of meteorological monitoring, and the data record on that goes back from that specific location on the hill in various places to the early 1990s, but you can kind of trace that data stream back to the early 1940s, I think, as far as McFarland Hill in general, in different places. We've had a weather station at Cadillac now for the past 3 years that has been collecting data year round, which is a first for us. So that gives us a little bit of a new perspective on there. Previously, we had seasonal weather data up there collected by Maine DEP in their air quality site, but that was always shut down after the ozone season was done. So it was discontinued in the winter, but now we have a rainwise station up there year round. And in the summer, we have data from Jordan Pond at the Jordan Pond House collected by the Friends of Acadia Jordan Pond Water Quality Project, which is the buoy project. And that's the only thing that we have run by the park. Now, if you go on Weather Underground, you know, you catch a lot of other little local sites in various places. But I'm not sure really who runs them or what kind of QA QC they have or durations of things. So there's probably a lot, but you know, it's pick and choose as far as what's accessible.
And
I was going to say, you know, my memory is not as good as it used to be, but during my time at Acadia, I do remember in the early 90s, Bill, when Bob Kohut was doing, you know, impacts of ozone on vegetation, that they were looking at different sites around the park to determine how ozone might be affecting plants. And there were microclimate clear microclimate differences. For example, I remember, I think it was near the Tarn, where there was a site looking at dogbane, and because, you know, there's I guess the impact of ozone on an individual plant species is dependent upon the amount of ozone available, the amount of moisture, and I think also, you know, the genetic susceptibility of that given plant. And for instance, at locations close to water, those plants showed more evidence of ozone injury on the plant tissue itself. Isn't that right, Bill?
Yeah, that's my recollection as well. You know, and I think some of those studies, I don't know if they utilize like small portable weather stations for, you know, at the various sites, or how they kind of amass the microclimate precipitation data, but I know a lot do, similar to what they're doing now with the Cadillac vegetation study. So there's probably a lot of really cool, very specific, site-specific information, but they're probably not all long-term, is my guess.
Nick, go for it.
Yeah, and thanks, Chris, for this question, which certainly we've talked about and others have as well. And I think the cool thing with Acadia is that it has some pretty interesting local climate gradients, which we've tried to utilize in some of our studies. As Catherine mentioned, the tree test bed study, which it's building on, actually off of Caitlin McDonough-McKenzie's PhD work, with 3 sites on Cadillac, a low elevation, a mid elevation, and a summit site, and then a 4th site at Schoodic. And between those four sites, it's an annual temperature range of 1.5 degrees Celsius. So not really big, but if you look at other measures, it's actually a huge climate gradient. So for July maximum temperature, it's a 5.6 degrees Celsius temperature gradient. For January minimum temperature, it's a 3.9 degrees Celsius temperature gradient. So there's some pretty big gradients. And looking at, for example, the number of hours of temperatures above 25 degrees Celsius, which can be really important for plants in their photosynthesis respiration, there's 8 times as many hours above 25 degrees Celsius at the low elevation site on Cadillac than there is over at Schoodic. So there's some pretty interesting big, you know, local gradients, and I think they're valuable to be able to sort of exploit, so to speak, to understand how plants are, and other organisms, and certainly in other systems as well, are responding to change.
And I'll just mention one other thing, that almost every research project now that happens in the park deploys temperature loggers. So we have temperature loggers like all over the place, but they're specific to individual projects, and those data typically don't get shared. But if anybody wants to take it on, it has been on my wish list for a long time, to organize a workshop to bring together people who are interested in sharing temperature data, to kind of figure out a format that people can easily do that. I think it would be really interesting, and I bet we could get pretty relatively fine scale data, at least over the past like 5 or 10 years, especially as hobos have become cheaper and whatnot. So anyhow, that would be an interesting thing and could easily result in a paper pretty fast, I would guess, if anybody wants to take it on.
Yeah, Abe, I'll volunteer to join the conversation. I don't know if I'll lead it.
You don't want to lead it, Jay?
Maybe if I can get some funding for it. But yeah, I'd underscore the value of the microclimate data. I've done a bunch of microclimate type work and trying to link it to long-term climate change. And there's that recent paper that came out in Science that looked at how the microclimate under the understory can actually result in an experienced climate change that's perhaps 50% more warming than the canopy experiences, because if you have a change in the structure of the forest and its buffering effect on the understory, because of the way the canopy forest responds to climate, that can cause the understory to feel like it's warming even faster than the overstory is. So I would second that. And I mean, there's challenges with trying to link up all these different sensors and different data sets and things that are a meter above the ground as opposed to 1.5 meters above the ground and stuff. For relatively cheap, if people are interested and want to put together a project, I'd be happy to be involved in it, where we do it right and do it once and deploy a huge network of sensors across the important gradients in Acadia and downscale a big climate model for the whole place. And it could be doable, but it's a big project, but it's definitely doable. There's some good examples of that. If everybody needs climate data, it seems like there's perhaps justification for doing that.
I'm in for the blanket of sensors, and yeah, we should talk more about that.
Cool.
And Hannah's adding that, yes, there's microclimate in the intertidal too.
It could be a cool project if you take it all the way into the ocean, and that could be a really, really good one.
Yeah.
I don't know if you get too wet though.
Yeah, no, I think that's a great project, a relative, famous last words, that is relatively straightforward, but it will be complex, but fun. So with that, I think we're gonna wrap up. We've got 2 minutes left. So I want to thank everybody for attending. This was great conversation, great presentations. I want to thank our team that helped to organize and run this and did far more work on this than I did. Emma Albee, who all of you know from email correspondence. She does a tremendous amount of work behind the scenes to support research in Acadia. Catherine Schmidt, who you've heard from a little bit today, and does a lot of the science communication. Roy Gott, who runs the IT and actually put together the Zoom bit and all of this. And thank you to all of the speakers and the folks that submitted posters and videos. And really thank you guys for attending and for all you do for the park. Our research community here is really strong. It's really exciting to be able to work with everybody. This year's been tough not being able to see everybody face to face very much, even during the field season. But the research that you do and the communication that you do is absolutely crucial to protecting Acadia's natural and cultural resources. And it's a big part of what makes this a special place to visit and work. So thank you. And our next session for those that want to come is going to be focused on early career opportunities. So if you're interested in hearing about early career, like internships, technician opportunities, seasonal opportunities, it'll be on Wednesday, January 27th from 12 to 2. And then starting next month and over the next few months, we're planning future sessions on adapting to the future, on a traditional ecological knowledge, on getting to know Acadia's visitors, and also the research permitting, reporting, data sharing, and science communication processes. So be on the lookout for those. Registration is open for the one that's later in this month, January 27th, on early career opportunities. Watch your email for registration on the others. The last note is to be looking out for email coming with the recording, for a link to the recording for this and kind of other information. And that will be your opportunity to let us know how we can make this better for you. Because really the Science Symposium is really for you, get you information and foster networking among the community. So with that, thanks a bunch. Really super appreciate it, and have a very good rest of the afternoon.
Thanks, everybody.